Bismillahirrahmanirrahim. Assalamu alaikum. I am Miss Marium Tariq. I am from Department of Zoology, Faculty of Biological Sciences, Qaid-e-Azam University, Islamabad. The topic of my presentation is the green approach for the synthesis of zinc oxide nanoparticles from Hedera, Nepal lenses leaf extract and evaluation of reproductive toxicity in the male rats. Abstract: zinc oxide nanoparticles are used in the production of various daily used materials including dyes, pigments, medical equipment, sunscreens, cosmetics, and healthcare products. The aim of our current study was to investigate the toxic potential of the zinc oxide nanoparticles on the reproductive system of the male rats. Zinc oxide nanoparticles prepared through the green synthesis using medicinal plant Hedera nipa lenses dissolved in different kind of organic solvents. For this purpose, adult male spark dolly rats were divided into 7 groups and were treated with different doses of the zinc oxide nanoparticles dissolved in distilled water, methanol, and ethanol, and given for 15 consecutive days via oral gavage. The results of present study showed that green synthesized zinc oxide nanoparticles from Hedera nipalensis leaf extract in the distilled water with the high dose, causing the pronounced reproductive toxicity towards the adult rat's testes, showing significant degenerative changes as compared to the zinc oxide nanoparticles in methanol and ethanol. It can be pre-concluded that this toxicity might be due to the poor capping of phytochemicals present in the leaf extract of the Hedera nipalensis. Then introduction. The nanotechnology is a rapidly developing concept in the various fields of science like medicine, agriculture, and physical sciences. Nanoparticles are the scale size particles with enhanced thermal conductivity and chemical stability due to their large surface area to volume ratio. The use of green approach for the nanoparticle synthesis is gaining importance due to its cost effectiveness, more catalytic activity, and reduced toxicity since plant extracted mediated biosynthesis of the nanoparticle is eco-friendly, economical, biocompatible, and offers natural capping agents in the form of proteins. Zinc oxide nanoparticles are involved in the reproductive toxicity. Zinc oxide nanoparticles accumulates in the cells and led to the cell death through oxidative stress. Zinc oxide nanoparticles were internalized by the testicular cells that resulted in the cytotoxicity in a time and dose dependent manner. Zinc oxide nanoparticles triggered the reactive oxidative species generation and alters the production of antioxidants or enzyme in the body. Methodology: first of all, we took the plant leaves, Hedera nipalensis, crushed them, weigh them, and form the solutions in different organic solvents like distilled water, methanol, and ethanol. And we grouped our spark dolly rats in different 7 groups. And first group is the control group, which is treated with only distilled water. Group 2 and 3 with the low and high doses dissolved in the distilled water. Group 4 and 5 with the low and high doses dissolved in the methanol, and group 6 and 7 with the low and high doses dissolved in the ethanol. And after 15 days, we dissect our animals and histology of testes, antioxidant assays, and testosterone levels will be checked. Results: significant decrease in the activity of cat, pod, and sod, and significant increase observed in the ROS and LPO in the testicular tissues exposed with the high doses of zinc oxide nanoparticles prepared in the distilled water as compared to the other groups. Significant reduction in the plasma and intratesticular testosterone concentration of the animals exposed with the high dose of zinc oxide nanoparticles in the distilled water. Histology of testes show this epithelial slowing off and wider lumen and less number of spermatids in the group treated with the high dose of zinc oxide nanoparticles dissolved in distilled water as compared to the other groups. The figure showing the control untreated group showing the normal testicular morphology and the other one showing the adverse effects on the testicular tissue when treated with these zinc oxide nanoparticles dissolved in the distilled water. Conclusion: the current study has demonstrated that zinc oxide nanoparticles has cytotoxic effect on the testicular tissue in a dose-dependent manner. Zinc oxide nanoparticles causes reduction in the testis weight, seminiferous tubule diameter, epithelial height, Leydig cell number, testosterone levels, oxidative stress by disturbing the antioxidant enzyme profile. Further investigations are needed at how nanoparticles penetrate the reproductive organs and cellular and molecular mechanisms of toxicity. The nanoparticle toxicity effects on testicular tissues will be useful in understanding the problems of male infertility. That's all about my presentation. Thank you for listening.